just want to point out again that everything that we'll be presenting will be here. If you go into workspaces and then BV-BRC workshop, under the AMR workshop, there'll be all these folders. Each folder has a different README in it that'll give you step throughs of what we do today. They'll have the job IDs, they'll have pictures, and they'll have the papers. But what I wanted to do briefly, because this is an AMR workshop, this particular study, which is the basis of everything we're going to do in the workshop today, this particular group, I think, took 250 samples from people who had had orthotic replacements, like a hip or a knee or a shoulder, and then they developed an infection. And when this happens, it's pretty serious. So they have to pull out the joint, so you're jointless for several months. And they were trying to figure out what were the bacteria associated with this that were causing this. So we're going to take one of those samples today and march through that. Spoiler, one of the organisms was Haemophilus influenzae. So we're going to drill into that because these were organisms that had antimicrobial resistance. Remember that anybody who has one of these surgeries, you're put on high doses of antibiotics because they really don't want this to happen. So they were resistant to it. So we're going to explore in this particular organism what genes are associated with antimicrobial resistance. And remember, this is a question I get a lot from people who are using the website, or a miscommunication, I guess, or they don't understand. Just because you have a blast hit to a particular gene does not mean that you have antibiotic resistance. We're going to dig in deep and look at individual mutations within those genes that are associated with resistance or susceptibility. So I just wanted to give us a quick background on different types of antimicrobial resistance. And there's a really excellent free course on Coursera, Antimicrobial Resistance Theories and Methods, that comes out of Denmark Technical University that I've really enjoyed, and it's quite a good course, and it's free. A lot of this I'm just stealing from them, so I gave them credit. For antimicrobial resistance, there are different ways that, well, if you're an antibiotic, that you can act on the organism. You can hit the cell wall, you can inhibit DNA synthesis, you can inhibit protein synthesis, folic acid synthesis, or osmotic integrity. And different antibiotics act in different ways. So the spectrum of activity is, this is just another one from one of their slides. So you can see what the range is. The penicillins, and ampicillin is a broad spectrum penicillin. It's been shown to work on gram-negative, gram-positive, and chlamydial organisms more narrowly than the sulfonamides. So, and the tetracyclines are the most broad, and some are more targeted. And so we're going to drill in specifically today on beta-lactam types. And the penicillin-based antibiotics often have beta-lactam. And ampicillin is one of those, and you can see there the range of things that they hit. And just so you know, that for the gram-negative and the gram-positives, what the difference is the way they build the cell walls. All of them have this lower cytoplasmic membrane with the periplasmic space and a bunch of these peptidoglycans lined up against each other, and some lipoproteins here. The gram-negatives have this outer membrane. And so for the antibiotic to work, it has to get through that outer membrane, either through a pore or some other way to get in there. Now, embedded in these things, and this is not exclusive that they're penicillin binding proteins, but when an antibiotic is able to get in and close, there are certain proteins that they can target. And penicillin has susceptibility to some of these in the penicillin types. Now, how do the penicillins work? They work exactly against this area, these peptidoglycans, they go in and disrupt it and prevent them from binding. And when the peptidoglycans are prevented from binding, you have a lot of the outside pressures coming in, whatever the aqueous solution is. And so it's starting to disrupt the osmolarity of the bacterial organism. And that's exactly how it's working. So it's inhibiting the peptidoglycan cell wall. And the way penicillins do this is this beta-lactam ring. So they have that, and that is what is binding to those peptidoglycans and disrupting the whole thing. And as the cell wall starts to be disrupted, everything moves in, it becomes a free-for-all. And finally, once the cell wall is finally breached, the bacterium is dead. That's when things go well for us. If you're the person who has this antibiotic, you really want this to work on that. But the bacteria fight back, and they have certain enzymes targeting these beta-lactam genes, which are beta-lactamase in the bacteria. So the ampicillin can get in, bind, and start disrupting, and then the bacteria starts producing these enzymes that are going to chop up this beta-lactam ring in the organism. So that's going to be the whole focus really of today's workshop, is this interaction. We're going to explore this very deeply with ampicillin-resistant organisms and look for mutations in a broad sense, in a very narrow sense, as to how this is working. The first thing a computer scientist is going to want to do, and we'll show you this how to do later, is to look at the reads. But when I have gone to the trouble of isolating an organism, paying for its sequence, the first thing I want to do is make sure that it's actually there and that something didn't go wrong, and maybe I've got contamination or something to do that. And so the tool we're going to start with first is taxonomic classification. So you go into Tools and Services and you click on Taxonomic Classification. Now, what you can do here is if you have your reads, or if you have an SRR number, a run number from the sequence read archive, which is how I found the example that we're going to work with today, you can just enter those numbers. However, if you need to upload things, when you click through it, through this little icon, so let's say you have some paired reads that you want to analyze, you can click on that and it'll take you to your workspace. But let's say I want to create a file right here. So I'm going to click on it, UVA April 2023. So I create that folder. And then to get to it, I have to do a couple clicks. There we go. Okay, and now I can upload files to that. And look at it here. What happens immediately when I click on that upload icon, it's already looking for reads. So it's important to have things tagged correctly. Often I get people saying, I uploaded stuff to my workspace and I'm trying to analyze my reads. And when I try to drill into it, I can't find it. That's because it's not tagged correctly. The pipeline needs to see this tag. If you're going specifically through the application, through the service, it will tag things correctly. If you're in your workspace, and I'll show you this in just a second, you may need to re-tag it if it was unspecified. And so I could say, select a file, and it's going to this computer, which is not my computer, so it doesn't have any files, but it would look for something that was reads, which is GZ or FASTA. And it says here, it'll look for these specific files. But we don't want to do that actually today, because we're going to use an SRR number. Going into the workspaces, BV-BRC workshop. Oh, good. Thank goodness. Over here into AMR workshop, and you guys should do this too, taxonomic classification. So I have the paper here, I have pictures, I have the README for this whole thing. But what I want to do is just to make it easier, because this is a text file, I want this number. This is a number at the sequence read archive. And if you want it to go, I'm going to open a tab to sequence, read, archive. And I could search for this number and you can see where it comes from. And how did I find these? In that paper, it lists a bio project, and then you can see all the samples associated with that. And I stepped through, I was just looking for a good one, and this one had a really good genome right away isolated in it, but you can step through those things. But here, once we have that number, this is so much easier. Used to, I had to download the reads to my computer and then upload them to BV-BRC, and it would take so much time and it was so frustrating. And now I can just do this. And once it's here, I move it over here and the reads are there. So I would encourage you to do the same. And then we have to go down here. So the parameters, what we use for taxonomic classification is Kraken-2. And Kraken-2 is looking at k-mers. It's a k-mer-based association, which are pieces of the genome. It's not looking at big things. I think they're like 12 mers. But it's looking through and it's associating different 12 mers with different taxonomic levels. And so it's going to give you a huge amount of things, but it'll show you in ranked order how many are mapping at a particular phyla, at a particular genus. So it'll at least tell me who's there. And that's the only one we have right now. And we're going to use all genomes. Now you can save the files that map to the Kraken database, or you can save the ones that didn't map if you wanted to explore those further. I actually never save anything at this point because we're going to show you FASTQ alignment service, where we're able to map things directly to specific genomes earlier. And that at least gives me more information. This would give me everything that mapped, which is a lot of stuff. So I'm going to be more targeted in my approach. I need to give it an output folder. Now, at this point, you can create a folder, but we've already done that. If you remember the name of it, you could type it in here, like if I did something for Brucella, which I work a lot with Brucella, but I'm not doing that today. So I don't want Brucella. But if I click on this down arrow, it will show me the things, or it should show me the things I most recently created. Let's see, I'll try UVA. Oh dear. Okay. I'll navigate to it. And then I navigated to the folder I wanted in. And Malik talked about giving things a specific name. Very important. You guys got to look at my workspace, which is not only what I do for my own research, I do it for all the testing for this website. And not only that, all these other guys dump their junk in my workspace because they're all doing their testing on my site, which is fine because we're all one big, somewhat happy family. So it's a lot of mess in there. So it's important for you to name it in a way that you understand. Think about it. When you're about to publish, you are stepping back probably a year or 6 months of what you did earlier, and you're trying to remember, what did I do? And you have to go in there. This happens to me all the time. I just was doing an analysis with these people that I sent them data 2 years ago, and they didn't get back to me. And then last week they said, we want to present this data at a meeting in 2 weeks. Moreover, we want you to do it. And like 2 years ago, I can't even remember anything from 2 years ago. So it's really important to name things correctly. And I keep telling you this, and I keep thinking I've solved the problem, but in this particular incident, I had to rerun the jobs all over again because I'd forgotten all about it. So I'm going to call this test, and then we're going to submit that job. This turns blue. When it turns blue, we have liftoff and we can move forward. So it's been submitted. You'll get a little message. Don't leave this page. Don't do anything until you see that it has been successfully submitted. There. Now we have the message that says It's been successfully submitted. So at this point, if you go away before then, it might not successfully submit, and you'll be very sad when you look for your job because you'll have to do it all over again. So once we have submitted it, you can go either through here up to the workspaces to see my jobs, or you can go here and it'll show you which jobs are queued, which are running, which are completed. This is generally where I go, and it's not there yet, but because it said submitted, I think it's just a slowness of the internet that's showing that.
Question.
Okay.
Can I submit shotgun data to?
Yeah, you can. Okay, this only does reads though. Well,
Yeah.
not only, look, because I use it for other things too. Taxonomic classification. You can also, by clicking here, bring in contigs. So if you have an assembly, I had the same people who've asked me to present this at a meeting also sent me some, they said this was packed bio data, but it was actually assembled contigs. And I ran it through this and I could see that it was highly contaminated. So I couldn't use that, but you could submit contigs too, because you can run an assembly algorithm, and we have assembly pipelines that we're going to talk about later today. So you can do that and you can submit these here as well to see what it is. Yes.
Were you asking in terms of the whole genome shortened metagenomic data versus the 16S RNA? Yeah, so this is, yeah, you can use genome.
You can also use 16S if you have it, but you can't assemble that data. Okay. Oh, look, now up here in the jobs page, it finally showed up. Okay, so it says that it's queued. And this is another thing. We get messages all the time saying, I submitted my assembly an hour ago and it's not ready yet. Well, first of all, assembly takes a while. So it's going to take maybe hours to assemble. Secondly, this is not your own private resource. We have 30,000 registered users and we have a queue. So you're in there with everybody else. We have programmed it to try to make it fair so that Thomas over here can't submit 800 jobs all at once because he's a whiz at command line and he dominates the queue. And the rest of the people like me who can only do one job at a time are booted back and we have to wait for his jobs to do it. So sometimes it takes time. If it's 24 hours and it hasn't come in, let me know. And then we look at it. And how do you let me know? You can click here. And in here, you can report the issue, which is here. And you could say, how come this is taking so long? And I often get things, seriously, I get this all the time. My dissertation is due tomorrow. I'm under tremendous pressure. Seriously, I get this stuff. And then you submit it and it submits a ticket that comes to me. And then I look at it and then I start ringing alarm bells right away. If it looks like there's a problem with the system, if it looks like it's just you have to wait your turn, I try to construct a gentle message to let you know that. If you've made a mistake and you want to kill the job, you can kill it here. Or, and this is really a good thing to have too, I can take a job that, let's say, I ran years ago, or I want to do it with a different assembler or some other parameter, and I can hit rerun, and it's showing me the job I did, and I can adjust it a bit. So those are good things that you can do. Okay, so we've looked over that, but let's look at this job. This job hasn't run yet, but if we go to BV-BRC workshop and we go to AMR workshop and we go to taxonomic classification, and here is a completed job that I have here in the workspace. When it's a job that's been completed, you can see that here I have a Word document. Here I have a text document. This is a PNG, which can be viewed, which is just showing you this image. And you can step back up there. And this is the completed job. It's got that checkered flag like you would see at Formula One or NASCAR races that cross the finish line. So I click on that and it gives me the information. This is the job ID. This is the start and end time. And if I click on the parameters, which is also very useful when it's a job you submitted 2 years ago and you can't remember what you did, you can click on the parameters and see that. And often when it's you guys writing to me and saying, this didn't work for me, I come in here and look at that to see exactly what you did. So I can see where the problems happened. We've got a report, the chart from the report, and these 2 text files that's showing you the total spectra of the hits that happened to this. This is the abbreviated version, which, believe me, is what you want, because the full report goes through every organism known to man pretty much, and it just goes on and never ends. But let's click on the report, and we could click up here for the view or here for the view. So it's showing you the reads here. It tells you use this guy. This is who you submitted. And here's the coverage. And this is the percent coverage. So, most of them were bacteria within the proteobacteria, and coming all the way here, it's trying to get as close as it can. But you can see that 90% of the reads are within this spectrum that drill into the Haemophilus. And then we have some that are Streptococcus. And then, never surprising, there's almost always some human contamination because people have handled these things. If you want to see how nice this looks, or have we're all primates and we're geared to look at things and see images of these things. So this is the Haemophilus is the majority. Here's the Streptococcus. Here's the Homo sapiens. And let's say I wanted to see what is about with these firmicutes. So I could click on that to expand it. Come on, let's expand. Oh, well, it didn't want to expand very much. Okay, I'll try the eukaryota. And then you can see that most of that was, Homo sapiens. And then you see, like, this is where it starts to get ridiculous. Apicomplexa, which are the things that like malaria and cryptosporidium go to, they probably share some chamers because they're eukaryotes with Homo sapiens, and people get misled by this all the time. So it's important to really think about how many of the reads are actually mapping here. So during the stories that we're telling, I do a lot of stuff with also endosymbionts, which are really small and they're embedded in cells of their host. And so I've been toying with ways I can get a complete genome, what size I would need to pull out and extract a complete genome from that. And so the levels that these guys are at, especially with eukaryotic genomes, just disregard that kind of stuff. Any questions on this? Yes.
We want to do the taxonomy of all of the different reads within one bio project. Is there a way to input all those SRA numbers without doing it
If you
individually?
wanted to.
But like without doing it individually and just finding every
Oh, if you could, you mean put in the bio project number and have them load all of that? We don't have
No.
that currently.
That's interesting though. We don't have that functionality right now, but I think that's something to think about.
But it wouldn't sort it out for you. So you get it all. And then it would be like, oh, well, which is with which? But you can't right now, but you're actually the second person who's asked about that.
Just a related point to that. So this version of the application that we have right now, it allows you to analyze kind of one sample at a time, right? There is a next version of this sample that we are currently working on. We have a developer and she is working on kind of next version of this application, which will allow you to use multiple samples as input at once. And we can probably think about using either bio project or something like that. And it will give you a comparative view of what is the taxonomic distribution on each of the sample, and give you a table where you can see what tax are over or underrepresented. So that's something that will be coming in the future.
Before I turn it over to Clark, I just want to point out that what we did today there in the README I can download it and open it, and you can see all the steps and the lovely little picture at the end. So that's all there for you to step through. Because you know what? You're going to be tired at the end of this and you're not going to remember everything. So this is there to help you.